Hi, welcome back. Today we're going to go through some chromosome anatomy and terminology. By the end of the video, hopefully we'll have a good understanding of each of these terms which describe the structure, ploidy relationships, and techniques used to investigate chromosomes. It's worth mentioning there are a few more techniques such as FISH and chromosomal microarray, but for the purpose of this video, I chose to just include karyotype to keep it central to chromosome structure. So let's start with the packaging of DNA. You'll note that DNA is a double helix, so two complementary strands of nucleotides wound around each other. To fit nearly 2 meters of DNA into a nucleus that's only a few micrometers wide, DNA is progressively packaged into higher order structures. So the hierarchy is the nucleotides at the bottom, the A's, the T's, the C's, and G's, which are aligned on complementary single-stranded DNA strands, which then wound together into the DNA helix. Now, first, the DNA wraps around histones, and these are proteins that act as spools. DNA and histones form a nucleosome, which is a basic unit of packaging. Nucleosomes then coil and compact further to a fiber called chromatin, and chromatin can be loosely or tightly packed depending on whether genes need to be active or silenced. During cell division, chromosome condenses even further into a visible chromosome, which you can see at the top. This is the most compact form, which is what we see in a karyotype. You'll see a karyotype towards the end of the video. Let's take a closer look at the chromosome structure at the top. This is what it looks like, and you want to be able to annotate each of these features. I'll go through each one, and then at the end, see if you can recognize them yourself. The telomeres are the protective caps at the ends of each arm made of repetitive DNA, which help maintain chromosome stability. And then we've got the arms. So the P arm is the short arm of each chromosome. You can remember P for petite being the shorter chromosome, sorry, the shorter arm, and then Q is the longer arm. Now, note that chromosomes don't always look exactly like this one, where the centromere is in the middle. Sorry, I actually don't think I mentioned the centromere. The centromere is the constriction point, so that circle in the center that holds the two sister chromatids together. So the two grey sides are the sister chromatids, and the centromere is holding them together. The centromere is really important because during cell division, this is where the spindle fibers attach to pull the sister chromatids apart. Now, as mentioned, it's not always central to the chromosome, so sometimes it can look a little bit different, and there's other ways. Telocentric is not in humans, so if we're studying humans, disregard that one. But the arms exist because where the centromeres sit on the chromosome. So, crucially, the centromere is rarely positioned exactly in the middle of the chromosome. So whatever DNA falls on either side of it becomes the two arms and they end up being different lengths. Remember P for petite, the short one, and then Q arms, the longer one. Based on the centromere position, the chromosomes in humans are classified into three types. So the acrocentric, submetacentric, and metacentric. Metacentric is where the centromere is near the middle, so the P and Q arms are roughly equal. Submetacentric is where the centromere is just off center, giving unequal arms. So we can obviously tell the difference between P and Q. And then acrocentric is very close to one end, so the P arm is tiny. This is what it looks like for chromosome 13, 14, 15, 21, and 22 in humans. Their P arms are so short that they mostly just carry ribosomal RNA genes. And then in other species, you can see a telocentric chromosome where the centromere is at the very tips. There's essentially no P arm at all. But the asymmetry in chromosomes is actually useful for chromosome identification in karyotyping. So when we investigate them. We want to be familiar with the terms chromatid and sister chromatid as well. Sister chromatids are the two that are joined at the centromere, and chromatids are the two that I've circled in red. A chromatid is one of two identical halves of a replicated chromosome. A single chromatid can be considered a chromosome once the centromere itself has divided, and chromatids are made of chromatin. Sister chromatids are just two that have been joined at the center with a centromere. Now, the terminology shifts depending on the stage of the cell cycle. So, if we're familiar with the cell cycle, before S phase, when they replicate, it's a chromosome. When we move from G2 phase to mitosis, two sister chromatids are going to be joined at the centromere. So these are the two duplicates that have been replicated in DNA synthesis. And then in anaphase, spindle fibers will attach to the centromere at the middle and separate the sister chromatids, which then each one becomes a chromosome again. And then moving on in meiosis 1, homologs pair and sister chromatids stay joined. In meiosis 2, the sister chromatids finally separate, and then we can create the haploid gametes at the end. Okay, let's talk about locus or loci and alleles. The locus is a specific position of a gene on a chromosome. So you can see I've sort of chosen two images in case one resonates more than the other. This one shows two loci. So loci is the plural of locus. Here we've got a locus for height, and here we've got a locus for being smoothed or wrinkly, like I don't know what that is, like a characteristic. Um, Here we've got a locus for color. An allele is a variant of a form of a gene at a given locus. So here we've got a gene for white flowers, and here we've got a gene for purple flowers. Here, both of these chromosomes have the same gene, so tall. If they were different, this one would say tall, this one would say short. Like at the bottom here, this gene has wrinkled and this one has smooth. So locus is kind of like you can think of it as the area. So that's one locus, or these are two loci. And then the allele is the variant form of a gene at a given locus. Now let's talk about ploidy. Diploid is a cell containing two copies of each chromosome. In humans, this is 46, and in all somatic cells. Haploid is a cell containing only one copy of each chromosome, so this is 23 in humans. And this state of ploidy is only present in gametes, so after meiosis 2. I've just included a visual here. So these are the gametes at the bottom, which are haploid, and these are the products of mitosis, which are diploid. And finally, here is a karyotype. Karyotypes are just a visual representation of genes that have been arrested in metaphase. So it's a technique used to visualize chromosomes under a microscope. And a karyotype displays chromosomes arranged in homologous pairs. We've got 23 in each person. 1 to 22 are called autosomes, and then the 23rd pair are sex chromosomes, which determine sex. In humans, each pair shows the maternal and paternal copy side by side. So here you've got mums and dads, or vice versa, we don't really know, but one copy from each parent. In humans, we've got 46 chromosomes, so 1, 2, 3, 4, 5, 6, and so on. 23 pairs, 1 to 23, and 1 to 22 are autosomes, and the last one is a sex chromosome. And that's it. I've got a summary here of what was spoken about for reference. Hopefully, this has given you a better understanding of the terminology used to describe chromosomes. And that's it. Thank you, and see you in the next video.